Glycolysis is likely the most important reaction in all of biochemistry. Every living organism, from the first bacteria to modern humans, uses glycolysis to produce energy. So, what is glycolysis? Glycolysis is the process by which glucose, or another 6-carbon sugar, is split into two 3-carbon sugar molecules. It occurs in a cell's cytosol, not any specific organelle. Unlike the rest of cellular respiration, it is anaerobic, meaning it does not require oxygen. It consists of 4 major steps: phosphorylation, lysis, oxidation, and ATP formation. Glycolysis produces, in the end, 2 molecules of pyruvate, the 3-carbon sugar, a net gain of 2 molecules of ATP, and 1 molecule of NADH. In eukaryotes, this process is accompanied by the link reaction, in which pyruvate is decarboxylated and oxidized to become acetyl-CoA, which is then used in the Krebs cycle.
The first stage of glycolysis is phosphorylation. The hexose sugar is phosphorylated by 2 molecules of ATP, meaning the ATP molecules each deposit a phosphate group onto the sugar. This results in a molecule called hexose biphosphate. Phosphorylation makes the molecules less stable and thus more reactive, and also prevents it from diffusing out of the cell. This leads us to the second stage, lysis. In the second stage, hexose biphosphate is split or lysed into 2 molecules of triose phosphate. Since the hexose biphosphate is unstable due to the phosphorylation, this happens spontaneously.
The third stage of glycolysis is oxidation. Hydrogen atoms are removed from the triose phosphate molecules. The resulting hydrogen ions are used to reduce the electron carrier molecule NAD, creating NADH.
The final stage of glycolysis is ATP formation. The lysis of hexose biphosphate and oxidation of the triose phosphates produces energy, which is used directly to make ATP. When ATP is made directly like this, it is called substrate-level phosphorylation. A total of 4 AT molecules are made per molecule of glucose, 2 per triose phosphate. However, since 2 ATP molecules are used in the first stage of phosphorylation, glycolysis produces a net gain of 2 ATP per glucose molecule.
In eukaryotes, glycolysis is followed by the link reaction. The link reaction is so named because it links the products of glycolysis with the Krebs cycle in the mitochondria. First, carrier proteins transport pyruvate, the final product of glycolysis, out of the cytoplasm and into the mitochondrial matrix. Next, the pyruvate is decarboxylated, meaning it loses a carbon atom. This lost atom joins with oxygen to form carbon dioxide, which diffuses out of the cell. The resulting 2-carbon compound is again oxidized, losing hydrogen molecules to reduce another molecule of NAD into NADH. This leaves an acetyl group on the 2-carbon molecule. Finally, the 2-carbon molecule combines with a molecule known as coenzyme A, producing acetyl coenzyme A, or just acetyl-CoA. As there are 2 pyruvate molecules produced per glucose, this process happens twice.
The story of the discovery of glycolysis provides an excellent case in point for the question of how can distinct pieces of knowledge from different knowledge communities be combined to create new knowledge. Initial investigations into the pathway began in the 1850s France, when the wine industry sought to understand why wine would sometimes turn distasteful instead of properly fermenting. Louis Pasteur showed that fermentation is done by yeasts, and the rate of fermentation decreases in aerobic conditions. In the 1890s, German scientist Edmund Buchner discovered that fermentation was possible using non-living yeast extract, because the extract contained the necessary enzymes. Later, in the 1910s, British scientist Arthur Harden and William Young discovered the glycolysis intermediate, hexose biphosphate. In the 1930s, another German, Otto Meyerhoff, linked all these discoveries together with the mechanisms of split hexose biphosphate into triose phosphate. Over nearly 100 years, different bits of knowledge were created through experiments and eventually linked together to form the knowledge of glycolysis we have today.
In summary, glycolysis is the important first stage of cellular respiration. Its 4 steps are phosphorylation, lysis, oxidation, and ATP formation. In eukaryotes, glycolysis is followed by the link reaction, the first part of the mitochondrial reactions. These 2 reactions produce a net of 2 ATP and 2 NADH molecules, which are later used in the oxidative phosphorylation stage of cellular respiration to produce more ATP for use by the cell.